strength of the flow of the direction of the circulation at that point in time. Okay. And this is just to give you an idea of the tropical storms in the Northeast Pacific at that time. Interestingly, Wally actually got down, I think it was an actual hurricane, but by the time it hit Hawaii, it was just a tropical storm, which is why the storm path, I think, stopped getting tracked by the National Weather Service. And then shortly thereafterwards, there was Genevieve, which came south of the islands. But it was a very active storm season because, as some of you may know, it was kind of like the El Nino that wasn't. Okay, so in general, now I'm cutting down that really long summer profile you saw only to basically from maybe like the day before Wally to before Isel. You can see that the general effects of Tropical Storm Wally was an increase in turbidity and a decrease in salinity. So there were about 6 inches of rain in a 24-hour period, so that's actually quite a bit. And then you can see that the stream responded really quickly. That's very typical of Hawaii because it has very steep, our geological islands, it's very steep mountains, and then the distance from the back of the mountains to the end of the watershed is quite short. The temperature changed, it recovered after 7 days. The salinity though, check this out. The salinity took 19 days to recover back up to its normal baseline levels, and the turbidity increased 257%. And what's interesting is after that, you can see that for some reason this area remains somewhat disturbed and much more sensitive to these bouts of just like these little showers of turbidityness. One thing that's interesting that you can't really tell from this data is that the storm surge was so great that it actually masked the tidal signal. After this happened, it actually caused quite a bit of damage to the fish pond. Pai Pai called us and said, what do you think we should do? And we recommended boarding up the Makaha to prevent a lot of water coming in, bringing with it a lot of turbidity. So let's see what happened when we told them to do that. So these two are managed storms because we advised them to close down or shut down the riverine inputs. You can see that there was still a dramatic wind event. The wind was at like 15.8 miles per hour. Of course, that's lower than a tropical storm, but given that Hawaii's trade winds are only 5 to 10 miles per hour, and you can also see it was in a very different direction. Normally, trade winds are like classic 80, maybe 77, on a 360 degree. This definitely changed directions. And we call Julio a temperature event because it definitely spiked up. The daily maximum temperature over that period of time, the fish pond itself was 34 degrees Celsius. And one of our students, Camilla, actually fell in the pond and she just was totally grossified because she was like, this is not a cool fish pond. It feels like a bath. Okay, so how did all of these changes affect the microbial dynamics? Okay, so I'm gonna tell you particularly about these stations right here. So this is just to frame for you. This is the area where we deployed the instruments, so we have matched physical and some geochemical data, and we have micros too. So what did we do? When I say we, I really mean my undergrads. Okay, so we took 1 liter water collections and we filtered it into 2 size fractions, and then we extracted DNA, and then we determined the density and abundance by qPCR, and then we also determined the diversity and composition, like what community is there, by doing 16S and then Illumina amplicon sequencing. And before I go on, I just want to, again, really give a shout out to the team. Of course, Kiana Frank she has really led the field efforts in my lab. This is Kili'i. He was our boatman who took us all around on our sampling, also aided by Paipai'oheia boatman Mari. So this is why they were absolutely essential, because look, they're holding these clipboards, which were like, can you hold this? And then can you read the YSI? They helped us with the YSI, the nutrient stuff, they helped us with everything. And then these are our amazing summer interns, Nicole Ferrant, Michaela Branco, she held our lab together. And then of course we had these wonderful summer interns, and then we had a senior scholar. And there's me, sometimes I was at the pond helping filter as well. Okay, so what did the data look like? What was the microbial diversity of the pond at this site, which we all call P7? So just to remind you in context, it's right here. And for the purposes, although I know it's a little bit contentious in terms of size class, we considered anything that was greater than 0.2 microns free living, and I'm saying it wrong. Anything smaller than between 0.2 microns and 0.45 free living, and anything larger than 0.5 particle associated, or something that might be with the sediment or with those things that was causing turbidity. And so what we're looking at here are those same time points, right before Wally, over the course of Wally. And so you can kind of think of this as what happened to the pond microbes and how did they recover in time in the free living versus in the particle associated. And in comparison, this is what happened during Isel and Julio. Okay, and this data, what we're calling out here, just some of the things that we think are kind of interesting. These are just representatives of some of what we've done here is we've attached popular names to large groups of bacteria. So something like, scary pathogens like Shigella, you can see that some of them came up a little bit more. Something like Rhodobacterialis is an alpha beta proteobacteria. Synechococcus and cyanobacteria, these are actually the really important bacteria in the ocean. These are their primary producers. How did they change? We can see that the flavobacteria, their populations definitely changed. Pretty much lots and lots of things changed. How do we compare that to the site right next door, P4? This site also changed a bunch. You can also see that it's a little bit closer to the more riverine inputs. And so you can imagine that it might get a lot more riverine influence from that. So you can see something like right post-Wally, the amount of Shigella that we picked up increased by like a whole bunch, for example. Whereas the flavobacteria dramatically changed, and a lot of the other general things changed as well. Okay. And then even as we get more inside and we look at some more of the things that are much more freshwater-like, we can see that a lot of the changes, so as we process in, the communities again change, but maybe they don't shift as much as they may have in these outer areas that might be, much more exposed to more tidal influences. And then we compare that to what an ocean riverine input is, and we can see that the community here looks quite different from the other samples that we looked at before, and potentially are dominated by much different organisms. Okay, so let's just look at, if we kind of use Danny's MATLAB program, and gave actually the data to Camilla, and we say, how does something like Synechococcus now change over the course of time that we were doing this so far? And so I believe this represents 30 weeks of data, would you say?
A third of a year.
Yeah, a third of a year. Okay, so that would be, I'm bad at doing math on when I'm standing up here. Okay, so it's a third of the year. So this is how something like Synechococcus, which is a really important picoplankton, would change over the course of time. Here's another. Oops. This is another one that we just pulled out. This is Oceanospiraliaceae. It associates with corals. It loves living in high salt and distributing corals, and you can see, so the warmer colors indicate the higher prevalence, so you can see it really comes in over the ocean breaks, and then kind of definitely, maybe, I don't even know if you want to call it a bloom, but it definitely is much more frequent in this area. So that's why I really like mapping these onto our little plots of our areas. Ultramonadales, it's a marine proteobacteria. It's not pathogenic, so you can see that sometimes our resolution is not so good if the quality of the data for that sampling point, if we lost it. But it's so interesting to me to see how the dynamics of these different bacteria change over the course of time period that we were sampling the pond. We even had a little bit of halobacteria growing. When halobacteria grows a lot, those blooms appear red. This is an example. This is actually an archaea, which is a whole other kingdom from most of the bacteria that we were looking at. And so you can see just how this community is kind of quite dynamic and can change over time.
Rosie, what is that picture in the lower left where it looks red? Is that
This is a lake that has had a halobacterial bloom.
Okay, that's not the fish pond.
No, this is not the fish pond.
Okay.
Yeah. And then, of course, because I used to be a bacterial pathogenesis, I said, Kiana, let's go look for the really scary stuff. So she said, okay. So she pulled out Shigella for me, and I was like, holy cow. Yeah, the Shigella, we have some, it actually does appear to change over the course of the experiment, and particularly seems to increase during the storm events. And so these are just different ways that we can look at how these kinds of bacteria, how they're how their frequency, their abundance really changes over the pond through time and through space. Of course, this is very important because the fish pond is actually used for food production, not only of fish, but they're trying to raise oysters here, and so we want to be able to inform the fish pond practitioners of when they should maybe not sell fish and other kinds of food products. So if we sum all the data, maybe, and look at larger groups, what can this tell us about how storms change the community bacteria, the microbial community, and how does that really influence what we tell the fish pond practitioners? How does this really help them become better stewards? So for that, we decided to look at certain groups of bacteria that have been used as indicators, more large scale in terms of water quality. So the Bacteroidetes. One thing we noticed, what this graph is showing you is a time course of just that area, that time period of from Wally to Isel and Julio, and then beyond to another storm event, Ana. So the idea was, if we have a signature of what a storm event is, can we then kind of look at our data in the future, maybe blind, and kind of pull out, that must have been a storm event. And we look at the 16S copies, we can kind of see that we do have an increase in the copy number of Bacteroidetes, which are human fecal indicators. This one particularly are human fecal indicators. During Wally, not so much Isel and Julio. Again, those were managed storms where the Makaha were boarded. So if you have input from the land, those were pretty much kind of more stopped. And then Ana, which was non-managed, again, a non-managed storm, you can see that you do have increases. And so it's color-coded so that high means more than 50 copies per mil, moderate 10 to 50 copies per mil, and low less than 10 copies per mil. And if we look at the general diversity, and we look at the Shannon index, so this is a general indicator of diversity, we can also see that microbial diversity also tends to increase a little bit in the storms. So, sorry, this actually should be shifted over here. This was like a conversion problem. But yeah, and then this is the Isel Julio, not so much. And then when we look at, again, another index, and when we look at the Chow indicator, we can also see that that Storm Wally was a major player in increasing the bacterial diversity of the fish pond at this period in time. Another way to look at this is to look at the beta diversity. So how might different kinds of communities that we're sampling at the same time, are they truly different? And so for this, we basically looked at what we're calling the particle-associated microbes versus the super free-living microbes. And so what's happened is this is colored, the free-living microbes are colored in red, and the particle-associated are colored in blue. And if you squint, you might be able to kind of see somewhat of a more clustering happening here. And so what is this graph? This is a graph looking at a lot of many different parameters having to do with the microbial community, and then graphing it in a in graphing it in kind of squashing those dimensions down into more simple space. When we look at those same kinds of samples, now just in the storms, we can see that whereas most of the samples that we have, cluster around this area, that the storms actually do cause a shift in the diversity of the microbial community more this way. So that's really interesting, in that we can actually pull out potentially how storms might shift the microbial community structure, and then because we have these weekly samplings, we can maybe look at succession of what happens to the microbial community after a storm. And how do these communities correlate to physical variables? So here we're looking at temperature, salinity, and pH, and I'm just going to start with the stuff that's not exciting. It doesn't really correlate with pH. It does seem to have pretty strong correlation, however, with salinity and temperature for the free-living stuff, and for the particle-associated ones, we seem to have a very strong correlation with temperature and salinity, and in this one, potentially strong correlation with pH.
What is the x-axis in those plots?
The x-axis in these plots, Kiana?
They're like kind of a random axis. They're just corresponding to the amount of an axis that describes a certain amount of variance of the microbial community. So in the graph before, she showed two different axes.
Right.
This is just one of them.
So is it a statistical measure of diversity, of microbial diversity?
Yeah, kind of. This is, well, yeah. Okay, we also, like I said, with the help of Pai Ohei'a, have nitrate, phosphate, and ammonia measurements. And this is less strong, I would say. I'm less convinced, except for the particle associated and its correlation with ammonia and potentially with nitrate. But the free living has, you can potentially make an argument for correlation here, but it's less strong. Okay, so what about the eukaryotes? And I have to say, I'm really interested in the eukaryotes because I just think they're really weird and awesome. So I said, what's going on with the eukaryotes? And maybe potentially because a lot of these eukaryotes, they are major players in the food web and can particularly either be taken up by grazers, which eventually are eaten by fish, or directly consumed by fish. So the thing with the larger photosynthetic microbes is that they might have really interesting trophic level interactions. Unfortunately, the eukaryotic cell density was not really affected by the storm. So you can see that there's, at least for our pico-eukaryotes, there really, and our other sizes, that there really was nothing to write home about as far as any of the storms. And I think that's also interesting and maybe speaks to our experimental design and something that we need to think about more. So the take-home message is there are a whole lot of microbes in the pond. It's quite diverse, and there's a whole bunch of them, which you might have been able to kind of suss out and guess for yourself. If you go there and you look at the pond, there's a lot of gunk in there. But what we're trying to start beginning to do is to establish a baseline of what does this fish pond look like. Because what we really want to be able to tell Pai is what a healthy fish pond looks like. Because they're restoring the pond back to what it was back when our kupuna were growing fish at higher levels than are now. And so we really want to be able to say, look, this isn't the ideal amount of nutrients. Obviously, there are many, many barriers that we're dealing with, including mangrove, including lots of different things involving climate change, and involving, I haven't even addressed, a lot of the upstream Mauka influence that might be affecting the fish pond. But one thing we can say is that what is going on in the fish pond might certainly be influenced. It's its own unique community. So this is Hei'a Fish Pond. Over the course of our study, during a storm, the community definitely changes, and its community is actually definitely different from, the wetland immediately upstream of this at Kakao'olivi. So Kiana has gone and even taken maybe some end member samples from the Hei'a wetland to kind of see, can we even see something from the wetland coming in? And the answer is yes, we can, but that community is still extraordinarily different. And that's likely because that habitat is extremely different. But of course, you could have hypothesized that, but we until now, we did not have the data to make those assumptions. And so we know that storms are significantly influencing microbes, they're increasing the diversity, they're increasing the abundance of pathogens for sure, and they're definitely changing the general overall community structure. Management also makes a difference. It might not necessarily change the diversity or the abundance of pathogens, or the community structure as much, but it might have very strong effects as far as things like the turbidity and the temperature. So in general, you should leave today knowing, if you didn't know already, that microbes are super important to this whole process and that they can definitely affect the nutrients that are in the pond, and ultimately, at least for the fish pond stewards, what is really important to them is how do they go about growing fish. And for me, what I would really like to do is be able to actually integrate different knowledge systems to create this baseline. So not only take, what we know about the microbial community and the physical factors, but I'm also, this is like work that I'm not talking about today, looking into different kinds of databases, Hawaiian language newspaper databases, looking at interviewing kupuna elders to kind of construct a theoretical idea of what a past ecosystem looked like, because that's really what restoration means, I think, is how can we get it back to a state, restore it back to a potentially healthier state. And in order to do that, I think it's extremely important to integrate multiple different knowledge systems. So like today, we integrated physical, geochemical, and microbial, and maybe later, Hawaiian language knowledge. Okay, so with that, that's all I have today, and I just want to thank you, and I'm open for questions.
When you were on your water quality samples, are Are the samples filtered, and to what degree are they filtered before you do the nutrient analysis?
The nutrient analysis samples are not filtered. No, they're just, yeah, they're just taken in one bottle grabs, we put them on coolers on ice, they're brought back to the garage at the Paipaio Heia, house, and they're just run right there.
So you
It's a
have no idea
colorimetric.
whether the nitrogen, for example, is dissolved in the water, in the bacteria, the cyanobacteria, or in the algae, or in the plankton,
Correct,
okay?
yeah.
Very basic question about the fish pond. Do you think the depth of the fish pond today is similar to what it was when it was actively used for food production? Or has there been a lot of siltation so
Oh,
that it's much
yeah.
shallower than it was?
I think it's shallower than it used to be. There are also areas that are extremely deep, and that might be, some gouging and erosion of the coral reef. So, I don't know if you remember, there's like a little island, so that island was actually formed as a result of the Keapuka flood. That island was the result of the floodwaters coming in, busting the wall through, and then forming that little mound, and then continuing on and busting through that away. And so that area is actually quite deep, even though there's sediment there. And so, yeah, we definitely think that the pond was not as heavily sedimented as it is today.
And so what's a typical depth today, and what do you think it was back in the day when the Hawaiians were fish farming it?
So at the very deepest, the pond is, I would say, 2 meters, all the way to 1 meter. Back in the day, I would probably say it wasn't much deeper. I mean, it was definitely photosynthetic all the way down to the benthos. But the difference is that the benthos wasn't sediment. It was probably more coral.
I'm just wondering whether restoration at some point might require removing a lot of the sediment.
Yeah, so the problem there is that you would need to have, EPA would have to approve that, because the huge concern there is how much turbidity, how much sediment is being released into Kaneohe Bay. And actually to rebuild the wall required huge permitting issues, which actually, because of our data set, we were able to provide Paipai the data that they needed to acquire the necessary permits, because we were able to show, like, okay, they've been removing mangrove all this time, this much sediment is normally released, it actually doesn't go on the bay, because it was really the Ruttenberg Lab's great efforts of sampling on the outside and the inside of the wall, so they were able to have those end member controls to prove that there would not be huge negative effects. Yeah, so the answer is, long term, they probably can't dredge the fish pond, which is ultimately what you need to do, which is what prompted that modeling study of how long would it take for the fish pond, all that sediment to naturally leave through circulation processes. There.
Did I understand? It was very fast for me, so my English is not my first language. So did I understand correctly that you were able to pick up human-specific bacteroidetes in your next-gen sequence database? You showed bacteroidetes. And
Yeah, we showed bacteroidetes. So actually, I won't say it was. I don't think it was human specific. It was not human specific. Or did we do? No, we did the PCR.
So we did the
qPCR.
qPCR, so that data was
That's right,
qPCR,
that
so
was
it was
qPCR.
quantitative.
Yeah.
I thought that's, yeah.
Yeah, it was qPCR. It wasn't picked up naturally by the amplicon sequencing. I mean, we knew there was bacteroides.
I don't know, I haven't looked.
Oh yeah, so
At that
oh
depth.
yeah, so I did look, it wasn't able to pick it up to the super specific level, but I knew that there were bacteroidetes and bacteroides there, and then we always knew that we were interested in looking at human fecal indicators, and so we built that into our experimental design, and that's right, Kiana did do the qPCR to test for
So
the human fecal.
Shigella was also qPCR?
Shigella wasn't.
No, that was just,
No, that was just based on sequence abundance.
yeah,
So it's more the sequence extended up and it's Shigella, not Escherichia genes.
yeah,
Yes, yeah.
yeah. Sorry.
Would you mind going back about 15 or 20 slides to those series of histograms that gave all the different colors for the different microbes?
Oh, yes,
Yes, these.
yes.
It strikes me, just initial inspection, the Shigella come and go like crazy here. I mean,
Yeah.
they dominate, and then the very next day or a few days later, week